announcements today are to remind you that quiz 4 is due on Sunday. I'll make it available on Thursday. Pick it up. Your books and notes and electronics away. Then pick it up. Then take it from around here and then turn it in Sunday night before 11:30 p.m. If there's a conflict because of Epic Expo or anything else coming up, let me know and we can work something out. As a reminder, there's no lab this Thursday. There is lab today, but there is no lab this Thursday because of no lab next Tuesday because of Epic Expo. And the schedule, I think, for Epic Expo has been revealed, or at least a tentative schedule has been released, so look it over, some interesting things, and get the most you can out of it. So, amazing, diverse set of academic explorations that you can get exposed to, including biology. Okay, today we are focusing, we're continuing endocrinology, obviously, and we're going to pick up where we left off regarding stress and then talk about metabolism. So we'll start with the glucocorticoids, like cortisol, primarily, touch on the catecholamines, epinephrine and norepinephrine, and then move on to other hormones related to metabolism and growth. So we'll move on to insulin, glucagon, and if there's time, thyroid hormones and growth hormones. So this was a slide that we talked about last week. The reminder is that stress, whether we're talking about just getting out of bed in the morning, or a bear charging at you, or a reminder that quiz 4 is due on Sunday, any of those are stressors, and those all trigger events in your brain that culminate in changes in your hypothalamus, and your hypothalamus releases CRH, a peptide hormone, into the portal circulation at the median eminence. CRH flows through the blood and diffuses into the anterior pituitary, where it binds to CRH receptors on corticotrophs, which are adapted to express those receptors and to synthesize POMC, which is metabolically proteolyzed to release ACTH into the systemic circulation. ACTH goes everywhere, but its primary target is the adrenal cortex, not the kidney. The adrenal cortex is a separate organ from the kidney, where it triggers the release of glucocorticoids, including cortisol. Glucocorticoids do many things, and one of the things that we want to focus on here with regard to metabolism is the changes in blood glucose. So it triggers blood glucose to go up. The glucose comes from the liver, a place where glucose is stored as glycogen, and from muscle, where glucose is stored as glycogen. The reason why we want to put glucose into your blood is for your brain. Your brain does not have stores of glycogen, but your brain is metabolically demanding. You need your brain to get out of bed in the morning. You need your brain to escape the bear charging at you. You need your brain to do the quiz 4. So your brain needs glucose, and it doesn't have its own sources. So we have the system for increasing blood glucose. Blood glucose goes to the brain, of course, and the blood glucose comes from liver and muscle. So here is a slightly more elaborate flowchart, stress, that we've talked about. Here is the HPA axis, stress, HPA. So right there is the HPA axis on this chart that we've just gone through. So I'm not going to do that again. Cortisol triggers these effects in the digestive tract. Glucose release from liver that I talked about, and we'll talk about glucagon and insulin later on in this class. Okay, so there's the HPA axis to release blood glucose. Stress also triggers your sympathetic nervous system to cause a change in heart rate, a change in blood pressure, and many other changes that we discuss in the other A and P class. Cortisol also has other effects that are important, but not focused on growth and metabolism. So if you're interested in the immune system, I don't know if Peter talks about cortisol inhibiting the immune system in immunology, but it's there. It acts as an anti-inflammatory drug, so inflammation is quelled by glucocorticoids, including drugs like prednisone, which is a glucocorticoid, it's a synthetic glucocorticoid, and it inhibits inflammation by decreasing the release of prostaglandins. Prostaglandins are an inflammatory paracrine signal. Okay, so this is the slide to focus on at least this part. The things in green are things to know for this course. Okay, so let's do the feed forward and feedback signaling. You actually answered a question like this on the quiz, so you should know the answers. What goes in these boxes? Anybody? Aiden.
Just out of curiosity, when was the names of the specific molecules involved in this stated in our previous slides or classes?
What's your point?
I was like, when I was doing this, I wasn't sure if you wanted, like, where we had the specific names for this. I don't think it was really offered.
So you're saying that I have to teach you everything there is to know? Even
I
if
was
it's
just
open book?
wondering if it was something that we needed to look up ourselves. Or
Yeah, it is something. It's open book.
Okay.
So this was on the quiz, and now we're following up. What goes in here? Anybody? CRH. And so CRH, and where is it released? Portal circulation, median eminence. What goes in here? ACTH. And what goes in here? If this is the portal circulation, what is this? Well, let's see what it is. Yeah, okay, so it's corticotropes are acting to release ACTH. And the effects of cortisol are like I said, cortisol gluconeogenesis, so synthesis of glucose, glycogenolysis, release of glucose from glycogen, and then glucagon we'll talk about later, and immune cells we won't talk about anymore. ACTH exerts short loop negative feedback, and cortisol exerts two forms of long loop negative feedback. Emily.
In the problem set, it talked about feed forward and feed control mechanisms. Is the short feedback loop and feed long feedback loop?
Right.
Is the short feedback one or feed forward?
No, the green is feed forward and the red is feedback. So this, you could say, well, I'm hearing that this was unfair to ask you about on the quiz because I hadn't taught, on the problem set, because I hadn't taught it to you, but I'm not convinced because it was open book. You could look it up, and this information is available in the book. And if you Googled feedback, if you just Googled CRH, ACTH, negative feedback, you'd get a figure like this.
But everything else was available on the slides, so it was hard to tell what you wanted.
Yeah, so 90% of So I taught you 95% of what you needed to know.
Just makes it more confusing for that 5%.
Okay. Confusing. I'll buy confusing. I don't plan to be, I never strive to achieve 100% clarity. I'm sorry. So a reminder that ACTH is a proteolytic fragment of a pro-hormone, pro-opiomelanocortin. A pro-hormone is a precursor. I wish I could say procursor, but there's no such word. A pro-hormone is a precursor to hormones. A pre-pro-hormone is a precursor. The pro-opiomelanocortin would have the signal sequence at the N-terminus. So pre-pro-opiomelanocortin has the signal sequence. Cleavage of the signal sequence in the ER, remember we're in the ER now, creates pro-opiomelanocortin. And then cleavage of pro-opiomelanocortin releases ACTH as well as other hormones, including melanocyte stimulating hormone and opioid hormones.
So if you cut out these specific hormones, do you still keep the N-terminus and the,
Well, if I only cut out ACTH, then I would have a fragment here that is the N-terminus, which is not a hormone because it's not completely cut to gamma MSH. But if I cut it here and here, now I have a fragment here, which is MSH, and I have this fragment, which is not a hormone, but is then recycled to make more POMC. Okay?
yes.
Yeah. So not all of it is hormone. This piece here is not a hormone. This piece here is. This piece here is not a hormone. This piece here is alpha MSH, which is a part of ACTH. So if we cut here and here, we get ACTH. If we cut here, here, and here, we get alpha MSH and a fragment that is not a hormone. Okay, let's use some of this knowledge to work on a problem. So I don't do this a lot. This is now pathophysiology, and I know this course is called anatomy and physiology, but let's use pathophysiology, disease, to understand physiology a little bit. So a disease called Addison's disease, which is also called adrenal insufficiency, which is also called primary hypocortisolism. Okay, let's unpack some of these words. Addison's disease, because of some scientists who characterize it. Adrenal insufficiency, because that describes the symptoms, syndrome. What does the word primary mean in this context? The word primary means that the defect is in the adrenal. It is hypocortisolism because the defect is adrenal. So you can imagine there would be secondary hypocortisolism due to a defect in the anterior pituitary, upstream of the adrenal. So we're not going to talk about secondary hypocortisolism, but any defect in the corticotropes that cause reduced release of ACTH would be considered secondary hypocortisolism. And any defect in the adrenal that leads to decreased release of cortisol is primary hypocortisolism. Remember, hypo means low. Yes?
What's about in hypothalamus, something like that? And hypothalamus, what about there?
So is there a tertiary hypocortisolism? I suppose there is. If the hypothalamus is not releasing CRH, you could get a tertiary hypocortisolism. I guess it must be pretty rare because I didn't find it anyway. So the symptoms of too low hypocortisolism, low cortisol syndrome. So the symptoms are fatigue and weakness, weight loss. These all make sense. Remember, cortisol is necessary to maintain metabolism, to maintain blood glucose. So these all seem like reasonable symptoms of signs and symptoms. Now, as long as we're talking about pathophysiology, just a sideline here, this is not going to be on any quiz or test. What's the difference between a sign and a symptom of a disease? There's a formal difference between a sign and a symptom. A sign is something that you can measure, and a symptom is something that is reported. Just so you know. So, these are the signs and symptoms of hypocortisolism. Now, I want to give you a problem that you can work on with your neighbors and try to figure out. Another sign of hypocortisolism is hypocortisolism is hyperpigmentation of the skin, darkening of the skin, especially in sensitive areas like around the neck, armpits, groin. Darkening of the skin, hyperpigmentation in sort of thin skins, thin parts of the skin, sensitive skins. So talk it out. I'm going to leave the room and go do something. There's something I forgot to do. So work it out and see if you can understand why low cortisol would lead to hyperpigmentation. You'll need to think about negative feedback. I'll give you another hint. You'll need to think about long loop negative feedback and synthesis of the hormones in the HPA axis.
Wait, hold on. Well, that makes sense because if you're sniffing out different pieces.
No.
So if you're sniffing out, if you're not sniffing out, there's negative inhibition in one pathway.
I have no idea. I'm very lost. What about you? I don't even know where to start with this,
I have
honestly.
two pairs of this one.
There was no way I was going to
Wow.
Like joggers.
or inhibiting the production of
Or,
HPA.
like, my initial thought is kind of like somehow it's affecting hypothalamus function. And so, like, it might not even be CRH, or it might be CRH or ACTH that is changing this.
Well, that's what's probably going to go back to
If you have less feedback, you can have more ACTH and more CRH, right? To get, like, a little bit of cortisol. So you have, like, a bunch of, I don't know, what do ACTH and CRH do? Do you know?
yeah, everything kind of does. Very rarely all in all.
Yeah.
Do you have the notes from that?
Which part?
ACTH and CRH, like what they do?
Yeah, so here's sort of an
Yeah. I think it'll be that whole.
CRH,
Shadow.
ACTH,
Cortisol can?
steroid hormones, or the other steroid hormones that are like melanin or something.
I'm not aware of cortisol changing. I mean, I never say never when it comes to hormones, but I'm not aware
Is it
of
even a hormone? No, I don't think it's a hormone, is it?
post-translation.
but it could evolve, it could go back and think that things
Okay.
Yes, it can do, well, because that's negative feedback. Yes, it certainly does that.
in that
I was
case.
gonna say, that sounds more like it.
Okay, so let's discuss this. Why do we have hyperpigmentation of areas of the skin in primary hypocortisolism? Somebody walk through the explanation of this sign. Do you have any courageous volunteers? John?
Weird thing has something to do with MSH.
It does have something to do with MSH, very good. So let's go, so first, what hypocortisol causes, it has everything to do with MSH. Why, is there increased or decreased MSH? There is increased MSH. Why is there increased MSH? Hypocortisolism.
That means it's positive, so there's less cortisol,
Less negative feedback.
Less cortisol, less negative feedback, hyperactivation of the less negative feedback is going to relieve the anterior pituitary of negative feedback. Less negative feedback. So the anterior pituitary is going to say, I need more. I need more. I need to do more in order to get more cortisol. Well, the anterior pituitary doesn't make cortisol, it makes ACTH. So the anterior pituitary is now getting stimulated to make more ACTH. Okay, we're not there yet, but now, go ahead.
So there's going to be less ACTH expressed, which means
More ACTH expressed.
More ACTH expressed.
More, and where does ACTH come from? What is the precursor of ACTH? POMC.
Oh, yeah.
POMC is the precursor for ACTH. So what the anterior pituitary does is say, okay, I need more ACTH, so I'm going to make more POMC, and I'm going to cut it to make ACTH. But in the process of cutting it to make ACTH, I'm also going to, byproduct, more gamma MSH and more alpha MSH. And I suppose more beta MSH. More of these MSH, melanocyte stimulating hormone. You have to know that melanocytes are the skin cells that give us pigmentation. So this was a challenge question because you had to put together loss of negative feedback, stimulation of the precursor synthesis in order to make more ACTH, but a side product, and you had to be able to translate melanocyte stimulating into pigmentation. So I would not ask you to do this on a test, but this is a way for you to put these pieces into a important clinical setting.
I would have more alpha MSH when you have acetylcholine, or not acetylcholine, ACTH, because wouldn't you be cutting ACTH and then you would have alpha just in ACTH and you wouldn't have a byproduct of that?
So it's trying to make more ACTH, but it doesn't have ultimate control over which enzymes it synthesizes. It's got the enzymes, they're there. So it's got the enzymes to make gamma MSH, it's got the enzymes to make beta MSH, it also has the enzymes to make alpha MSH, which presumably is not very active, because what we're trying to do is make cortisol. Remember, enzymes don't have to be in high concentrations to be able to work if we give them plenty of substrate. Yeah,
But isn't this cleavage, because if we assume that there will be enzymes like in various concentrations, as long as POMC concentration is high, it's gonna do all the cleavages.
that's right.
On POMC, I think then we can never make like beta MSH or alpha MSH, because we will always make ACTH or the opposite. And so,
Well,
wouldn't it be, I feel
it's
like.
kinetically controlled.
Right, and so if it's kinetically controlled, I feel like with this pathway, it will make more ACTH, but it would only cause the cleavage of PMC at the ACTH side, but not at the other,
Not this one.
not the gamma or any other substrates, it would only make ACTH because the pathway is telling it to make more ACTH and not the other
The
hormone.
pathway is telling it to make more POMC.
So how is it to be specific to make ACTH?
It's never specific to make ACTH. Whenever ACTH is made, other hormones are made. Now, there could be control. I mean, I guess I want to say it's never highly specific, right? All we have to do is decrease the expression of the hormones, of the enzymes necessary to do this, and increase the expression of the enzymes necessary to do this in order to make more ACTH relative to anything else. And we can do that, we do that, but there's always some expression of enzymes, and enzymes can be active if you give it a substrate. So that's why I say we almost always make these other hormones when we make ACTH. Is that clear?
And would you argue it's in the, like in certain areas of the skin, as you said, like neck or armpits, because the concentration would be low, that would only affect like thin skin?
Yeah, so the
compared to others?
effect of MSH is most obvious in certain parts of the skin that are most sensitive to getting stimulated, having their melanocytes stimulated by MSH. So they're the parts of the skin that, I mean, obviously this would be difficult to detect in a dark-skinned person. So this is a sign that is really only valid for light-skinned people. So, because this effect would be obscured in someone with dark skin. I'm not sure it would be invisible, but I think it would be more difficult to detect in someone of African or South Indian or Pacific Island ancestry.
If someone's like stressed a lot, will they get darker skin over time? Like, will that happen, or is that just not enough?
It will happen. It's slow. It's a slow effect because you have to stimulate the cells to proliferate and manifest darkening. So it's a slower effect than some of the others. What are the causes of primary adrenal insufficiency? Well, there can be an autoimmune disease where the immune system starts to go haywire and recognizes the cells of the adrenal cortex as foreign to be attacked. So this is an autoimmune destruction of cells in the adrenal cortex. Certain infections can trigger immune and other negative effects on the adrenal cortex, or there can be a genetic error in the enzymes necessary to make cortisol, right? A genetic defect in one of the, I'd have to pull up the slide on the cortisol synthesis, but a genetic defect in one of those synthesis enzymes could cause primary hypocortisolism. There is also a syndrome called Cushing's disease, which is hypercortisolism. And this, I'm just going to describe this for interest, but primary hypercortisolism Cushing's disease, again, named for some scientist, medical scientist who characterized it, is elevated cortisol, either due to a benign tumor of the pituitary or a benign tumor of the adrenal, hyperplasia of the cells of the pituitary or hyperplasia of the cells of the adrenal, higher production of ACTH, leading to higher production of cortisol, or just normal ACTH, but high production of cortisol. And it has some strange ish signs and symptoms. Obesity, fat deposits in the neck and shoulder, and face, giving rise to a moon face. It's described in the literature as a moon face, big, round, puffy face. Slow growth, acne, pain, weakness. I will not test you on symptoms and signs. I might test you on language, like what does hyper and hypo mean, but, or not just test you, but use it and expect you to be able to understand. Yeah?
You said that adenomas are tumors?
Adenoma is anything with an oma at the end is a tumor.
Okay, so should we think of like cancer tumors?
Yeah, these are cancers.
Okay.
Adenomas are not usually fatal tumors. These are benign tumors, but they can have effects if they're causing too much release of a hormone. They have effects, but they aren't the kind of cancer that becomes metastatic and invasive and kills you.
Okay.
All right, so now we're going to move on to metabolism and then growth. Okay, so let's move on. Any questions about the physiology and pathophysiology and the tools that I'm trying to give you to interpret physiology and pathophysiology? Before I go on, I do want to make it clear, the quiz is anatomy of the leg and of the endocrine system. It is not physiology. And the key concepts list that I gave you that has blanks that you should be filling in, and the list of the leg muscles that you should be learning about are the target of the quiz. The physiology that I'm talking about here is going to be on the next exam. All right, let's talk about insulin. Insulin is a peptide hormone. That fact is quizzible, because that's the anatomy of insulin. Okay? Its stimulus, the stimulus is high blood glucose, like after you eat a meal. When a typical person eats a meal, they digest the food, food is converted into glucose, glucose is absorbed into the blood in the digestive tract. That's something we talk about in some detail in AMP2. But it should be pretty clear that high blood glucose happens after a meal. And that triggers cells, called the beta cells, in the pancreas to release insulin. So, insulin being a peptide, and insulin being released from the cells, the beta cells of the pancreas, is anatomy. The target cells of insulin are liver, muscle, and fat cells. Now you may think fat as being just, fat, but it's actually cells. Adipocytes are the cells that make and store globules of fat. So they're cells, they respond to stimuli, and they make or release products of fat. They make fat or they release fat into the bloodstream depending on the body's needs. So they're fat cells. So the targets of insulin, liver, muscle, and fat, that's anatomy. I'm just trying to be clear about what is and what is not covered on the quiz. The receptor is a tyrosine kinase. I'm not going to test you on receptors on the quiz. Tyrosine kinase. So you should, as soon as I say that, receptor tyrosine kinase, you should be thinking binding, dimerization, just like you did on the problem set. Binding, dimerization, cross-phosphorylation, recruitment of scaffolding proteins, RAS, and then a kinase cascade. Okay, that should just come at your fingertips from receptor tyrosine kinase. Insulin is a peptide. So it's got an N-terminus and it's got a C-terminus. This is actually pro-insulin because it's got the signal sequence and it hasn't been processed. So let's walk through the processing. Pro-insulin is synthesized by the ribosome attached to the ER, and this protein is being fed through the translocon. I hope you can see the image in my mind as I say these words, because the image should be in your mind of the mRNA interacting with the ribosome interacting with the ER, translocon, and then this being co-translationally translated. Cutting of the signal by the signal peptidase releases proinsulin into the lumen of the ER that is then also proteolytically processed and disulfide bonded. So disulfides are formed and proteolysis happens to make the final peptide hormone, insulin. And what does insulin do? Well, it causes the liver, muscle, and fat cells to take up glucose from the meal and store it for when you're starving. Takes up glucose uptake, synthesis of glycogen, the storage starch for glucose. Take up amino acids if your meal happens to have protein in it. Let's absorb those amino acids. Let's synthesize protein as a way of using the goodies that we just ate. Remember, we evolved in a world where you never knew where your next meal was coming from. You couldn't count on it. So every meal was precious to store as much of those nutrients and calories as possible. And storage of fatty acids from the fat that you ate into triglycerides in your adipocytes, your fat cells. So insulin is critical for storing the products of digestion in liver, muscle, and fat. Let's talk about the mechanism. So now we're talking physiology, clearly not part of the quiz. The mechanisms of glucose-stimulated insulin release. So what cell are we talking about here? We're talking about a pancreatic beta cell. Pancreatic beta cell. These are the cells that detect glucose and respond by releasing insulin. These are the source cells of insulin, and their trigger is high glucose from the meal. Glucose in the extracellular fluid that's in contact with blood from the meal. Glucose goes through a uniporter. GLUT2 is a uniporter, a facilitated diffusion transporter. Using the language from cell physiology, I'm trying to reinforce a concept. A uniporter. Glucose diffuses through the uniporter, not through the membrane. It's not simple diffusion, it's facilitated diffusion. It's uniported into the cell. Okay, what happens here? What goes in this blue cloud that is obviously covering something? What do we do with glucose? glycolysis, and then, what? Oh, there's something between, we do, but what's between glycolysis and oxidative phosphorylation? The citric acid cycle, we metabolize glucose. And I'm trying to remind you of some self-is concepts, right, that I don't talk about in A&P very much, but it's still really important. Glycolysis, pyruvate oxidation, citric acid cycle, NADH, these should all be ringing bells, crazy. NADH, electron transport chain. I know it might be painful for some of you, but it's still true. NADH, electron transport chain, proton gradient, oxidative phosphorylation. What's the bottom line of all of that stuff that I just said? ATP. So what I just said is summarized into glycolysis, and in this slide they just called it respiration, but it's missing a few things that you should be able to decorate the slide with more information than just this. But the point is, it increases ATP. So it increases the ratio of ATP to ADP. Right? More ATP is going to put more in the numerator, so this number, this ratio is going to go up. But the main point is that ATP goes up. Now I'm going to introduce a new channel that I haven't introduced before, because this is really only very important in the pancreatic beta cell. An ATP, it's called the ATP sensitive potassium channel, but a more descriptive name would be the ATP blocked potassium channel. This is a potassium selective channel that is blocked by ATP. Okay? So now you know that ATP has gone up, and there is such a thing, an ATP blocked potassium channel. What goes in this box? What goes in this block? What happens when we block a potassium channel? Depolarization. Then, I'll give you a hint. We have voltage-gated channels. What do you suppose are the most important channel that we need to introduce to get to hormone release? Calcium channels. So depolarization, what goes in this block? Activation of voltage-gated calcium channels. Calcium influx, T-snare, V-snare, synaptotagmin story, release of hormone. So this is a beautiful, I love this story, because it combines so much of what you've been spending this year studying. In a fairly simple story. You should be able to understand every single step in this, and maybe even more than shown on this slide. Any questions about the molecular mechanisms of glucose-dependent insulin release by pancreatic beta cells? No?
So the ATP sensitive potassium channel, does it let in potassium?
Which way does potassium flow through a potassium channel? Out. The concentration of potassium is high on the inside and low on the outside. That you needed to know that last semester, and you needed to know it now. High concentration of potassium, low concentration of potassium. Potassium flows out. Flowing out of a cation leads to less negative inside. Or, I'm sorry, less flowing of potassium out, because we're blocking it. So flowing of potassium out is what made the negative resting membrane potential. Less of that outflow, less negative on the inside, less negative is a depolarization. You had to be able to take those steps.
Sorry,
Right.
then what does it let out?
It lets out potassium.
Oh, sorry, yeah,
So the channel lets out potassium, and the ATP prevents it.
right. That's, okay.
Right? Okay, so now let's talk about the molecular mechanisms of insulin-dependent changes in fat, liver, and muscle cells, right? Now we're taking it, insulin has been released into the blood, but we need to talk about muscle and liver, and you can annotate your slide, fat as well. Insulin binds to insulin receptors, which are receptor tyrosine kinases. This slide is pretty weak. Signal transduction, blah. I mean, let's unpack that. What happens when insulin binds to the insulin receptor? Dimerization, conformational change, cross-phosphorylation of the subunits, recruitment of adapter or scaffolding proteins, including what's the one that I want you to have the name of? RAS. Activation of the kinase cascade or MAP kinase cascade. That's what is embedded in this word signal transduction. The kinase cascade leads to changes in the cell, including changes in gene expression, changes in synthesis, in the metabolic synthesis of the fat and muscle cells to increase glycogen synthesis and fat synthesis and protein synthesis. It also increases the expression of a glucose uniporter that brings in the glucose to feed these synthetic pathways. I will not ask you to know the difference between GLUT2 and GLUT4. I don't care about that. I don't mind reading that you know the difference, and I won't take points off if you get it backwards, but it's not a bit of knowledge that I expect you to commit to memory.
I'm asking you, do you mind to share with us now what is the main
Oh.
difference between GLUT4 and GLUT2?
The main difference is that GLUT2 is expressed in pancreatic beta cells, and GLUT4 is expressed in fat, liver, and muscle cells. Other than that, they're pretty much the same. Yes?
So, do you want us to specifically name RAS with the receptor tyrosine?
Yeah, I do.
story?
Yeah, that's an important part of that story.
Okay.
Laura?
So, you lost me a little bit, so after RAS is recruited,
After RAS is recruited by the adapter,
correct?
it is activated by its binding to those proteins.
Okay, so they cross-phosphorylate each other
Yeah.
after the conformational change, which recruits RAS, right?
And other things.
And other things. And then after that happens, that is the signal transduction?
RAS activates the MAP kinase cascade. So now we have activation of MAP KKK, MAP K, MAP kinase. Remember that this is cell-phys. MAP kinase is a kinase that phosphorylates many things, including transcription factors, as well as enzymes, to activate the enzymes or to activate changes in transcription. MAP kinase does a lot. So you can decorate this slide right in here in this white space with all that information that is embedded in this simple phrase. So what are the missing steps? I should have done this. Cross, there it is, right? That's what I just said. John?
So is the phosphorylation of GLUT4 what allows glucose to enter?
That's a good bet. I'm not positive that that's true, but I would bet money on that. That one of the substrates of MAP kinase is GLUT4. That increases both the function, but it also probably increases the expression of GLUT4. So it increases both its function and the number of GLUT4 proteins in the surface of those cells. Okay, let's do glucagon, and then we'll take a break. So glucagon is sort of the antagonistic hormone to insulin. Insulin and glucagon are like the biceps and the triceps I'm sorry, insulin and glucagon are to blood glucose what the biceps and the triceps are to flexion or extension of the elbow. They work against each other. And we need both. I mean, we need this constructive antagonism in order to control blood glucose. So glucagon is a peptide. It's synthesized as a precursor called pre-glucagon, but it's not there is no pro-glucagon, but that's not a detail I expect you to know. It's a peptide hormone. That is anatomy that I want you to know. Its stimulus is low blood glucose. It is released by the alpha cells in the pancreas. Different cells, but in the same gland, the pancreas. That's anatomy. Its target cells are also liver, muscle, and fat. Its receptor is a GPCR. That is not a fact that well, that's not going to be on the quiz, let me just say that. It turns out it's linked to GS, and so you should immediately be running off with the GS story. You wrote it on your problem set, you've got it in your mind, you should be able to just plug in that cassette and play it. It does the opposite of what insulin does. It breaks down glycogen, it breaks down lipids, it decreases glucose uptake in liver and fat so that whatever glucose we've got is going to the brain that needs it. It decreases amino acid uptake, it decreases protein synthesis. It's trying to conserve blood glucose because you don't want to starve the brain. It's a peptide, that's all this is trying to illustrate. So let's take a break, and I want to talk about this antagonistic control after the break. That I teach in both AMP classes. And the concept is antagonistic control. So the idea is, well, this slide is pretty unhelpful, so let's go to the next slide. Antagonistic control of room temperature. We have a heater and we have an air conditioner. Those are going on right now. There's a heater unit somewhere in this building that can pump warm air into this room, and there's an air conditioner somewhere in this building that can pump cool air into this room. And there's a thermostat that sets a set point. If the temperature goes low, the power to the heater is increased, and the power to the cooler is decreased. Adding power to the heater increases the temperature, getting it closer to the set point. If the temperature goes high because the sun comes blasting in, or all our body heat is being collected, the temperature goes high for some other reason, the power control unit sends less power to the heater and more power to the air conditioner, which brings the temperature down. So you know about antagonistic control if you just think about thermostats and heaters and coolers and a set point where the power to the heater and the cooler are the same. And by power, I don't mean watts or anything, I just mean the effect of the heater and the effect of the cooler are now completely counterbalanced, and we are at a stable set point. But in fact, what probably happens in this room is that the temperature goes up a little bit and then comes down, and then maybe it overshoots, and it probably cycles around the set point. Goes up and then comes down, goes low and then goes up, goes up and then comes down, goes low and then comes up. We probably can't detect it, but there's probably a cycle around the set point. We do the same things in our body. We have a set point for serum glucose. This is a typical non-diabetic set point. If the blood glucose goes low, let's do high first. We eat a meal, and we pump a bunch of glucose into our bloodstream. That causes stimulus of insulin release, by the mechanisms that I talked about a few minutes ago, and inhibits glucagon release by mechanisms that I haven't talked about. Insulin triggers uptake of glucose, so the blood glucose goes down. Now let's say you're starving, but your brain still wants glucose, needs glucose. So if blood glucose goes down, we decrease the synthesis and release of insulin from the beta cells, and we increase the synthesis and release of glucagon from alpha cells, and that liberates glucose from the fat, muscle, and fat storage areas of our body, and brings the serum glucose up. Insulin and glucagon have an antagonistic control to set a set point of serum glucose, about 1 mg per mil. Normal. In diabetes, which is a complicated disease, and I don't have time to describe it, we have abnormalities in either insulin or glucagon. Remember, glucagon is not glucose, and it's not glycogen. You've got 3 important G words here that you have to discriminate. Glucagon, glycogen, and glucose. Don't get them mixed up. If you say, just don't get them mixed up. Well, I don't want to give you an example of mixing them up because that'll sink into your brain and you'll mix them up. So diabetes is an abnormality in insulin secretion. That's usually juvenile onset primary diabetes. Or it's a disease of insulin reception, which is usually adult onset secondary diabetes. But it creates abnormalities in blood glucose, typically high blood glucose. Yes?
Sorry, are those the different types? That like type 3, type 2?
Type 1 diabetes is a defect in insulin secretion, and type 2 diabetes is a defect in insulin reception. Okay, so antagonistic control. Okay, we're switching gears big time now. We're talking about growth. So we're transitioning, not immediately, but pretty soon, from metabolism, control of blood glucose, to control of growth. And growth is a complicated thing. Your bones grow, your muscles grow, your brain grows, your organs grow. Growth is a complicated phenomenon. And we're not going to talk a lot about the physiology of growth. We just don't have time for it. So we're lumping it all into the concept of growth. But remember, most of the time, the normal physiological period of growth is up through puberty. And then growth, then we reach a period of homeostasis where our body size is stable, at least, and for most of us, it stays stable for quite a few years, and then as we age, you can either have weight gain or weight loss, depending. So let's talk about thyroid hormones. Thyroid hormones are tyrosine derivatives. They are amines. They are iodinated amines. That's anatomy. That's the chemical anatomy of thyroid hormone. The stimulus is for the thyroid gland to release thyroid hormone is TSH. The stimulus from the thyrotrophs of the anterior pituitary. The stimulus for thyrotrophs of the anterior pituitary to release TSH is TRH from the hypothalamus into the portal circulation. Yeah.
So is the release site for TSH from the pituitary, or is it from the body?
The release site for TSH is released from the anterior pituitary into the systemic blood, where it circulates to the thyroid, and triggers thyroid hormone synthesis and release.
So the target is the thyroid.
The target of what?
The target of TSH.
The target of TSH is the thyroid.
And
The
TRH.
target of TRH are the thyrotrophs of the anterior pituitary. TRH is released from the hypothalamus, and it triggers the thyrotrophs of the anterior pituitary to release TSH, which triggers the thyroid hormone, thyroid gland, to release thyroid hormones. So this, maybe you read this one in backwards to read the story. Let me say that again. TRH from the hypothalamus, released into the portal circulation, triggers the thyrotrophs of the anterior pituitary to release TSH into the systemic circulation, which triggers the thyroid glands to synthesize and release T3 and T4, the thyroid hormones, into the systemic circulation. That has pleiotropic effects. So, I'm not going to ask you to know the name of the cells in the thyroid that release thyroid hormone. They're in the thyroid gland. They have thyroid hormone has widespread effects. So TRH has a specific effect, TSH has a specific effect, but T3 and T4 have ubiquitous, multiple effects. Is that clear? The receptor for thyroid hormone is an intracellular receptor. So now you should be off and running with the story. Carrier protein, diffusion through the membrane, diffusion through the cytoplasm, binding to an intracellular receptor, conformational change, transport to the nucleus, changes in gene transcription. In this case, it's an upregulation of gene transcription, but that's not always true for all intracellular receptors. Sometimes they're a negative transcription regulator. The effects in the target cells, liver, kidney, hearts, brain, muscle, is to increase cellular respiration that we've talked about just a few minutes ago. It increases glycolysis. It increases the enzymes of the Krebs cycle. It increases the production of mitochondria and increases mitochondrial production of ATP. It increases the activity of the sodium, the expression, and therefore the activity of the sodium potassium ATPase. It pumps up metabolism in order for cells to live and survive and, in some cases, divide and grow. Where is the thyroid gland? Thank you. Okay, so this is key information, both anatomy and physiology, and you should be able to tell which is which on the thyroid hormone. Yeah.
Since the T3 and T4 receptor complex in the nucleus, it binds to transcription factor and actually upregulates it. And so here the effect is always upregulation of genes.
In this case.
Transcription, correct?
The transcription effects of T3 and T4 binding to its intracellular receptor is an increase in transcription.
So as long as if it inhibits the transcription.
Not in this case.
Not in this case, but generally, if it inhibits the transcription factor, that's when you'll see a downregulation
That's
of
right.
transcription.
Okay, but I guess the point is there can be inhibition of transcription. Sometimes that leads to a stimulation of cells, because if the inhibition of transcription of an inhibitory protein happens, now you have increase in cellular activity because of decreased expression of an inhibitor. That can happen. So now let's go through the synthesis and release of thyroid hormone. So this one I gave you, I think it's a little out of order on your handout, but I gave it to you big because the print was too small if I printed it small. And let me dim the lights. I'll put you at risk of falling asleep, but it makes it easier to see the slide. Okay, capillary. Blood. what hormone is in this blood that is going to stimulate this thyroid cell to release T3 and T4. So that TSH. TSH is in the blood, and it interacts with a TSH receptor. And frankly, I can't remember what type of receptor TSH is. If one of you want to look up on my slide from one of the previous classes, you can find it. Or Google TSH receptor. I think it's a G protein. I think it's GS coupled, but I'm not sure. Anyway, TSH interacts with a receptor. In the thyroid cells, the follicle cells of the thyroid, there's a protein. The thyroid cells make a protein called thyroglobulin. That's what TG stands for, thyroglobulin. Thyroglobulin is a protein, and it has tyrosines. Those tyrosines, the thyroid gland also has enzymes that are iodotransferases. They take iodine from your blood, or from the cell, cytoplasm, or from the blood that you eat. You eat iodine. Iodine is an essential nutrient. Sometimes you'll see in the salt that you buy, it says, contains iodine or doesn't contain iodine. Typically in our diets, we have plenty of iodine. We have more iodine than we need. But iodine is essential here. The iodotransferases take iodide, the iodine ion, iodide, and transfer it onto the tyrosines of thyroglobulin. Then there are other transferases, MIT and DIT. Well, no, MIT is monoiodotyrosine and DIT is diiodotyrosine. And anyway, we're making iodotyrosines with an enzyme, iodotransferase. So, and we're actually making this two-ringed structure. So we iodinized tyrosine, and then we transfer one iodinated tyrosine to another one, and make this two tyrosine thing. It's got a name, but I don't even know it. Two tyrosines still attached to thyroglobulin. This is happening in it's called the colloid. The follicle has a sort of a gooey center. The follicle cell surrounds a gooey core, and the gooey core contains thyroglobulin and enzymes to make this two tyrosine ring structure iodinated in the colloid, or the lumen of the thyroid's organ, thyroid gland. This is endocytosis, so clathrin-dependent endocytosis. That should be triggering more self-is-dependent memory. Endocytosis of the thyroglobulin and lysosomes. There's actually two ways of going through here. This endocytosis and lysosome and proteolytic cleavage releases T3 and T4 from thyroglobulin. So we store thyroglobulin. The thyroid gland stores thyroglobulin, but it doesn't store T3 and T4. Okay? The thyroid gland stores thyroglobulin, but doesn't store T3 and T4. It synthesizes T3 and T4 in response to TSH, which isn't shown here. TSH triggers the processing of thyroglobulin. Why is that? Because this is one of the stories where T3 and T4 are not released by a calcium synaptotagmin snare-dependent process. As soon as we cut this thing off of thyroglobulin, it's lipid soluble, and it just goes wherever it wants to go, including into the blood, where it can interact with carrier proteins that carry it all over the body. So the point here is that TSH stimulates the cutting of T3 and T4 from thyroglobulin, and the release is not a calcium-dependent story. Because T3 and T4 are lipid soluble. Aiden?
So what's MIT?
Monoiodotyrosine.
And so what's happening first? I mean, aren't these all sort of happening at the same time?
Yeah, this is all happening at the same time, creating this double ring structured modified thyroglobulin in the colloid of the thyroid gland.
And so I know you have like, it looks here as though these iodine structures
These iodine structures, so this
are
is,
forming sort of right next to the lumen, but then you have stuff kind of further away from the lumen, further down, when you're looking at sort of how TG is being reused.
all of this is happening in the lumen.
So, okay, so it's just,
All the iodination and then the transfer of one iodotyrosine to another iodotyrosine, making this dual iodotyrosine structure. This is all happening in the lumen of the thyroid.
okay.
So once T3 and T4 are released, what happens to the thyroid gland?
Oh, it probably gets recirculated, recycled.
So the
and-
whole point of this is to release T3 and T4.
The whole point of this is to store this this double iodotyrosine structure, and then to make it available for release of T3 and T4. Yeah, yeah.
Sorry, then what does it not store? You said,
It doesn't store T3 and T4. It stores thyroglobulin.
Wait,
And everything's happening in the lumen.
Thyroglobulin is in the lumen. T3 and T4 are released in the cytoplasm of
Which is
the,
the next one.
and then it goes everywhere. It just diffuses, including into the blood. And remember, this space here is exaggerated. This capillary is right up next to this follicle cell.
So there's still a T snare and B snare over here.
There's no T snare and V snare in this
How
story.
does the endocyte, doesn't you have vesicle release though? A vesicle fusion?
Oh, I guess, okay, I'll buy that this processing of this clathrin coating thing and then fusion of these vesicles, that could be snare dependent, but it's not calcium dependent. That's constitutive. Yes?
is TG just the couple ring? Or is
TG
the entire
is,
thing?
TG is, this thing is TG that has been modified.
Okay.
Thyroglobulin is the protein that contains iodotyrosine.
And they're both TG, the top one and the bottom one.
Yeah, I think we would call them both TG.
So which one's the actual hormone then?
This is the actual hormone. T3 and T4 is the actual hormone.
Both of them?
Yes, T3 and T4 are both hormones.
They have different functions.
No, they don't. The body converts T4 into T3. T3 is the most functional thyroid hormone, but they both have function. So, okay, I'm not gonna rush through this. So, I would like you to think about this for homework for Thursday. Look up the word goiter and think about why primary hypothyroidism could cause goiter and why hyperthyroidism could cause goiter. Goiter is a sign of both hypothyroid and hyperthyroid. Think about that and see if you can make sense of that. And that's where we'll start on Thursday.